Good morning, guys. Welcome to the first lesson of Module 7. For this lesson, it's just a brief overview into studying genetics, studying inheritance, and recapping lots of stuff that you will have done at GCSE. So throughout this lesson, I'm going to be giving you some time to pause the video and complete some tasks and then replay again when you're ready. Likewise, because it's a video and not a live lesson, you can stop at any time, rewind at any time, and listen to me over and over and over again. Lucky you. Now, to start with, we're going to take a look at genetics as a basic concept and quickly recap the key terms from GCSE. As you can see, down in the bottom corner, there's only 19 slides, and lots of those are me just waffling some information at you. So hopefully this shouldn't take too long, because the next couple of lessons are quite meaty, and there'll be a video for those, again, uploaded at a later date, probably on Dropbox as well, because these are quite big. So to start with, pause me for a second, have a look at the picture, tell me what it shows, how you can describe those pictures, and can you tell me anything about them? Pause now. Okay, hopefully you should have had some ideas about what the picture shows. We're not going to go over what you should have got just yet, because it's an introduction lesson and the recap will come later. So, the title for this lesson is Studying Inheritance. We're just going to be looking at the keywords. As you can see at the top, in some of these PowerPoints, there are links to boxes. And these are where they might link to previous learning that you've done, but also previous content covered in your A-level. So this one specifically links to Module 4, looking at behaviour of chromosomes during meiosis, so the production of gametes, and also mutation, so specifically looking at how genes are changed during the process of cell division. Now, hopefully, if you're my class, then this will be on our OneNote class notebook. If you're with Miss Goldie, this will be hopefully somewhere. There's loads and loads and loads and loads of keywords for this topic. Okay, unfortunately, that is the way it is, and we can't really do anything about that. But they are key for this topic. You need to be able to apply the keywords as best as you can. I'm just trying to find the file on my computer. Bear with. No. Admin. Resources. Keywords. Right, so for Module 7, just so you're aware, there are what appears to be 4 pages of keywords. 4 pages of keywords. It's going to be fun. It's a challenging topic, I'm not going to lie to you. It is quite difficult, but it's the kind of thing, like much like maths, once you know how to do it, you can do it. Although knowing my class from year 13, none of you do maths, so this should be fun. Now we're going to take a look at those keywords that you will have learned at GCSE. Okay, genotype and phenotype. Now, unfortunately, at A-level, we start to ramp up the difficulty a little bit, and now we need to know that the genotype is the combination of alleles within an organism. Okay, and alleles, remember, are 2 or more versions of the same gene. Okay, and this is, or the genotype, and your genotype specifically, sort of limits how much of something you can be. Okay, so those of you that know me know that I'm quite small. Obviously, my genotype has not enabled me to reach the lofty heights of 6 foot tall. Okay, my genotype is actually limiting me to being the height I am. Okay, so the example here in blue, you could grow to be 1.9 meters tall due to your genetics. Maybe you've got some really tall parents. Maybe you've got gigantism. I think that's a thing. Rocking on in your family line. But if your environment means that you don't have the necessary nutrients and things like that to help you grow, you're not going to get there. Okay, so the genotypes are specifically there to determine the upper limits or the lower limits of an organism's characteristics. Now, we should be confident in linking genotype to the next word that we're going to look on. So if you're not too sure, pause now, have a look over this, look at your books, because we're going to move on to phenotype. Okay, so the genotype causes the phenotype. Pheno, if you imagine that PH has an F sound, sounds a little bit like face. It's what you see. Okay, the observable, biochemical, because remember we're looking at internal characteristics as well. Okay, so observable is to do with external, generally. Biochemical, the internal characteristics of an organism. Now, sometimes our environment can be entirely or wholly responsible for altering someone's phenotype. Okay, so I might dye my hair bright ginger. I might have tattoos. I might decide that I fancy a little bit of fake tan. I don't know why I would, but some of you might do. Okay, but also things like scars. They are permanent changes to your phenotype that are not caused by your genes. Okay, so it's important to remember that the genotype affects the phenotype, but the environment can also affect the phenotype without the impact for genes. Now, could you please have a go? Pause me now for maybe a few minutes. You need to write these sentences out using what you know, using your noggins, maybe using Google if you've got no idea what's going on, and take a look at these sentences. Okay, fill the gaps, use your keywords, get that knowledge. Pause me now. All right, so hopefully you've paused and you haven't skipped on in the video to find the answers because, you know, I don't trust some of you as far as I can throw you. But this is where your gaps should have been, or this is how your gaps should have been filled. So a sequence of nucleotides bases makes a length of DNA. Remember, our nucleotides were found in Module 1. They are made of a pentose sugar, ribose, deoxyribose. If we're talking about DNA, it's deoxyribose. We have got a nitrogenous base. Remember, that is A, T, C, or G. Adenine, thymine, cytosine, or guanine. And we have also got a phosphate group. Okay, and then we've got a phosphate group. Okay, just up here. Now, hopefully, this should be fairly easy for you to recognise. Now, if these nucleotides are bound together, we've got our phosphodiester bonds between the phosphate of one nucleotide, I'm just going to write nuke, and the ribose, or the sugar, or the deoxyribose, should I say, of the second nucleotide. Now, if you think back to your structure of DNA, we've got a 5' because it's bound to the 5th carbon on this ribose phosphate, and we bind our nucleotides in a 5 to 3 direction, 5' to 3' direction. Okay. Now, in our DNA, those combinations of A, T, C, and G, those nucleotide bases, have a specific sequence, and in some sequences, we call them genes. Okay, they code for proteins, polypeptides, if you're being fancy and specific, which we are because we're scientists, which can either be structural, so maybe giving things like collagen, or functional, such as enzymes. Okay, so remember that a gene, or we've got DNA, which turns into RNA through transcription, and that turns into a protein through translation, and that gives us either the structural components to make us human, or functional components to help us inside. Thinking back to GCSE, genes that have 2 or more forms are called alleles. You've got the dominant and the recessive that we're going to take a look at in a second. And the place where a gene is found on DNA is called a locus. So if you think back to the starter, we had these chromosomes, we had little sticks, and they had things like P's, and there was maybe an R down at a different point. Those locations are the gene's locus. The plural of them is loci. Okay, so like octopus, octopi, locus, loci. Now, if we think about us in terms of genetics and going back to that GCSE when you did mitosis and meiosis again, our cells are diploid. Okay, we say that we've got 46 chromosomes, but they're 23 pairs. Okay, so some of you might have seen the karyotype picture where they've got like 1, 2, 3, and basically there's just lots of squiggles all lined up. Okay, those pictures of those chromosomes, the diploid means that there's 2 copies of each chromosome. We get that diploid genotype from 2 haploid or half copy gametes. Okay, so you know that a sperm has 23 chromosomes in it, and an egg also has 23. Now, hopefully, even if you don't do maths A level, you should be able to do 23 plus 23 and gives you 46, which is, as I said, what we've got in those cells. Now, for A level, we've got a little bit more language, and this is a horrible word to say and to spell. The 2 copies of the same chromosome are called a homologous pair, or they're called homologous chromosomes. Sometimes you might see them as homologs. Okay, homo meaning same, like homosexual or homo something else. Not homo sapiens though, that's a completely different thing. Okay, now the important thing to recognise and look at this picture is that each chromosome has the same gene locus location. Okay, so if this is the chromosome, if the purple one on the left is from the mum and the pink one on the right is from the dad, you'll notice that gene A is found on the same position down the length of the chromosome in each of those chromosomes. They don't mix around and jiggle about and just hope for the best. That's where they are. Now, thinking back to your GCSEs, hopefully these words should look fairly familiar because they weren't that long ago and you actually did the exam. So hopefully some of that knowledge should still be in your brain. Homo, homozygous. Oh, that could have been a better example for using last slide than homosexual, but here we are. Homozygous is when you've got 2 copies of the same allele. So for the example, we've got 2 big A's. Whereas heterozygous, like heterosexual, is where you've got 2 different alleles. Notice that I'm using the word alleles and not gene because alleles are different versions of the same gene. So we can have dominant or recessive. If you're not too sure, pause now, maybe recap GCSE. I've heard Free Science Lessons guy on YouTube, it's very good. And then carry on because we're going to take a little bit of a look now at what those 2 words, dominant and recessive, mean. So the dominant and the recessive alleles, I'm just going to skip through this. You can pause if you wanted to read the information that I've just said because I speak quite quickly, but I'm going to get straight into it. Dominant alleles are the powerful ones. Okay, they're big. We write them in capitals. Okay, so if in my genotype I have got a dominant allele, I'm going to see that characteristic. So for example, if A or big A means that I've got curly hair and little a means that I've got straight hair, regardless of if I've got 2 big A's as my genotype or a big A, little a, I'm still going to have curly hair. Okay, because it overpowers the recessive gene. Now there is a difference at A level, and this is maybe a few lessons down the line, probably in a couple of weeks. We have got something called gene masking. Okay, now gene masking is where the presence of one gene sort of overlaps or completely obliterates the presence of another. But we'll worry about that when we get to it. Conversely, recessive alleles are always written in lowercase, so it might be 2 little a's, for example. And the phenotype is only seen if you are homozygous recessive. Okay, now when we're talking about homozygosity, okay, homozygous recessive, you have to be very specific about which gene you're saying is homozygous. So you're either homozygous dominant or homozygous recessive. Okay, and in this case, if I was homozygous recessive for my example, I would have straight hair because I've got no dominant allele to completely override that. So pause now, have a go at describing the genotypes for each of those combinations of alleles below. So you've got 1, 2, 3, 4. If you would like to, you can use the example of hair in the key that I've got at the top, and you can describe what hair type they might have as well. Okay, so pause me now, and then we'll move on to the next slide. For this example, the person on the left-hand side of the diagram, number 1, is homozygous dominant. Okay, they would have curly hair. You would write their genotype big A, big A. The middle two are heterozygous. They've got one each of a big A and a little a, and they would therefore have curly hair as well because they've got that dominant allele, that big A, that gives them the curly hairedness. Over on the right hand side, we are saying that they are homozygous. I'm just going to write homozyg because I can't be bothered. Recessive, because there's no capital letters there. We write their genotype as 2 little a's, and they would have straight hair. Okay, nice and easy, hopefully, because it's going to get a little bit harder in the next couple of lessons. But in some situations, the combination of alleles being heterozygous doesn't lead to just the dominant phenotype being produced. And that's called codominance. So imagine I've got a white cow, this one, and I've got a red cow, this one. If I combine those two mummy and daddy cows, the offspring that they have are spotty. Okay? This is called codominance. We've got a whole lesson on it, so don't worry about it for now. But this is where both alleles occur together. So generally, the offspring are heterozygous and the phenotypes become blended. You can say that they're both manifested. And you can see in this cow down here, it is white with brown spots. Or is it brown with white spots? Who knows? I think there's more white than brown, so I'm going to say it's white with brown spots. Okay, but again, there's a whole lesson coming on it, so chill your beans for now. Just know that it exists. Now, the final thing is thinking about what happens if there's more than 2 alleles. Okay, so previously at GCSE, you were told to make sure the allele definition was 2 or more. This time for blood type, we've got something called a multi-allelic trait. And this is when a gene has more than 2 alleles. Okay, now, just because we've got more than 2 alleles, i.e. maybe blood type A, blood type B, and blood type O, we can only express 2 of them. Okay, remember that's because we've only got 2 copies of each chromosome. So regardless of whether I really wanted an O allele, if I've got 2 A's, doesn't matter. Okay? So multi-allelic genes are when there's more than 2 possible letters, combinations that are going to give us our phenotype. Now this is going back to the starter, okay? At the very beginning, you might remember that I asked you to tell me what you knew about this picture, how can you describe it, and can you tell me anything about it? Hopefully now, through the last few slides, you will be able to add a bit more detail. So could you please draw out those pictures of chromosomes? You're going to pause me, and you're going to add as much detail as you can. Just to give you a few hints, there's a couple of arrows already on this picture, but you are welcome to add any more that you see fit. So pause now, scroll through when you're ready, and we'll go through some of the answers that you might have had. Now, for this slide, you can see that we've got lots of answers. We've got homologs being mentioned. Notice that they've put a UE at the end. I think that looks a little bit ugly. OG is fine. They are homologous chromosomes. So the diagram has mentioned that they're a pair of homologous chromosomes. One is inherited from the dad or the male parent, and the other is from the female parent or the mum. Just in case you weren't sure, maternal means from the mum, and paternal means from the dad. Okay. Now going down the diagram, we can see that they've labelled up locus, and what it means, it's the location of a particular gene on the chromosome. And at each locus, an individual has 2 alleles, one on each of those homologous chromosomes. You might also have mentioned the different genotypes that there are for each of those genes, A, B and C, and maybe you will have flagged up that there's 3 different pairs of genes at 3 different loci. Important to recognise that because they're different genes, they're not all going to be found at the same place. If they were, that would be silly. Okay, it would be very crowded and it probably wouldn't work very well. You'd probably have all sorts of issues. So if you didn't get any of those, or you added any more detail, maybe if you're in my class, put it in the collaboration space on our OneNote Drive thing. I don't know, I'm making up how to do the technology. Collaboration space is good though. Or you can send me an email if you're not too sure whether things are relevant there. Now finally, I would like you to have a look at either page 156 if you've got the Year 2 Oxford book, the blue one, or it's also on page 420 if you've got the Year 1 and 2 book. If you haven't got either, I'll take a photo, and if you're in my class, because I think it's more likely to affect you, I will add it to our lesson resources, class resources bit on the OneNote folder. Could you please, with that image, diagram, whatever you've got available, have a go at making your own version of the flowchart to show the links between keywords. Okay, remember, we are looking to integrate ideas. It's a synoptic course, so we want to try and bring things and show how they link together as easily as possible. When you are done, have a go at the summary questions that I will attach to my class. So if you are in my class, we're going to upload the images and stuff that you might need to the content library for Module 7, and it will be under Lesson 1, and I'll put the pictures and stuff there. Otherwise, if you're in Miss Goldie's class, maybe send her a little email if she doesn't give it to you already or you don't have the books, and ask for them. So thanks very much, guys. Hopefully you found this useful. I will also upload some exam questions for my class just to practice using keywords and things like that. But otherwise, this lesson is done. You can stop the video now, go and recap anything, maybe make some notes and get your keywords up to scratch, and I'll see you for the next one.